Thank you very much. It's great to be joining everyone today. Just as an introduction, I'm Courtney Noah, Vice President of Scientific Affairs at BioIVT. And today I'm going to be talking about a foundational, but what I think is often underestimated driver of success as it relates to new approach methodologies or NAMs. And that is that the quality and selection of your starting material really is what dictates the downstream biology and biological success of your assays. So we spend a lot of time optimizing platforms, assays, and analytics. But what we consistently see here at BioIVT is if that starting biology going in isn't right, everything downstream can become noise. So today I'll focus on how sourcing, characterization, and of course, quality control of human biospecimens can really drive improvements across reproducibility, increase that confidence, and ultimately support regulatory acceptance of NAMs. So let's start with a quick overview of BioIVT. BioIVT is strongly positioned to support your biospecimen requirements at any stage, whether that's from target all the way through to clinic. And just to take a moment to go through our portfolio areas, as you can see, we have healthy and disease state clinically collected specimens. Those can include both tissue as well as biofluids. We have human and animal matrices that are typically used as control materials. We have our immune cell products and services portfolio. That includes collection of our leukopaks through our BioIVT donor centers, and of course, our ADME products and contract research services. But let's get back to the focus of our discussion today. So when we look at translational failures, especially when we're looking at complex modality development, we tend to attribute those failures to efficacy or safety issues. But you can really trace those failures back and see that they often originate much earlier. We see issues that can be driven by poor translation of non-human systems. Sometimes those targets that we're looking for aren't actually relevant across the patient populations that we're moving forward in clinical trials. And of course, variability that isn't completely understood or perhaps controlled for. So across all of these, there's a common theme that the biology and models weren't aligned to the right question being asked. And that's exactly the problem NAMs are trying to solve. But as you can imagine, NAMs still depend on the quality of their inputs. So NAMs are designed to be more predictive and human relevant, at least that's their purpose. But that only holds true if the input biology actually reflects human disease. If your specimens are poorly characterized or perhaps inconsistently collected or not representative of the patient populations, you can start to introduce bias, variability, and definitely uncertainty. So the question becomes, are we enabling NAMs with the right biology or are we just replacing the model? And there's a very real cost to getting this wrong. Studies have shown that the majority of researchers question their findings or limit their work due to biospecimen quality issues. And that's just not just a scientific risk, it's an operational risk. And when the sample fails, you lose not only that sample, but the time, the reagents, and potentially weeks of progress that's been made in that research workflow or program. So improving that starting material quality isn't just about better science, it's about reducing the risk across the entire development pipeline. To address this, you really need to think about quality as a system and not just a checkpoint. High quality starting materials must rely on these four key components. And just to walk you through them, of course, standardized collection protocols, making sure those SOPs and documentation are in place. Controlled processing, leading to control over those preclinical variables, really making sure that those are minimized as much as possible. Ensuring that you have detailed clinical annotation. That's critical information to have as you're trying to compare across different studies. And then that rigorous quality control, because quality isn't something that you should just verify at the end. It really defines the biology from the very beginning. And so the more rigor you put up front, the less variability that you're going to start to carry forward into your NAM system. one of the most critical and underestimated factors is donor variability. So, what we have observed through the many thousands of collections that we've done through donors at our donor centers is that variability between donors is significantly higher than variability within the same donor over time. So, in this example, we're looking at immunophenotyping data from our repeat leukopak collections, like I mentioned, done through our BioIVT donor centers. And there's stark differences that we see between the immune cell compositions when we're looking between donors versus within repeat collections from the same donor. And what that means is that the donor itself is not just a source of material. You have to really consider them as that primary experimental variable. So when you change donors, you're not just introducing noise, you're really changing the biology that's driving your system. And this can have major implications for that assay reproducibility, model development, and of course, translation into clinical settings. So, how do we manage that variability without losing human relevance? Because that's what we're trying to drive towards, right, with NAMs. And this is in the context of immunology-based studies, but I think you'll see parallels to some other research applications. So there are three key strategies. First, using recallable donors will help you ensure that consistency across studies. I understand not always possible, but where it is possible, it's really important that you're looking to that to try to drive some of that consistency in your assays. Second, look for the appropriate donor characterization. In this case, where we're looking at an ADCC assay, we may be important to look at phenotypic or genetic profiling, such as HLA or FC receptor status. It really is dependent on the assay in question. And third, make sure that you design your cohort strategies intentionally. So variability is something that you're controlling and not something that's just going to add additional noise to your data. So this really allows you to strike that balance between the reproducibility, but also incorporate that real world biological diversity, which you have to include for it to be truly relevant and down the line predictive. And so, like I mentioned, that's critical for optimizing NAM performance and, of course, improving that translational relevance. Quality control also needs to be fit for purpose. And for example, if you're working with solid tissue, what could look like just a single tumor is actually very heterogeneous in many cases. So a resected lung tumor, for example, may get divided into multiple blocks for processing. And with that, each block can contain very different biology. So one may have that true adenocarcinoma, while others could have fibrosis, inflammation, or necrotic tissue. And what we know is that the clinical diagnosis alone won't tell you what's within each of those blocks. So the risk is real here. If the wrong block is selected, you could be measuring gene expression or target presence in the wrong tissue context entirely. And what is that going to do? That's going to lead to false target validation, and of course, poor translation as you work further downstream. So this is where that pathologist review, that QC step with the pathologist review becomes really critical. By confirming the diagnosis at the block level and quantifying that tumor percentage, necrosis, and immune infiltration, we're able to ensure that the biology you're measuring actually matches your scientific question. And at BioIVT, every tissue specimen goes through this board certified pathologist review. And what that translates to for our customers, it's reducing that variability, it improves data accuracy, and gives them the confidence that results truly reflect disease biology and aren't just artifacts of the sample. And I love this stat here, and this is why not compromising on quality standards is so important. At BioIVT, we actually maintain a less than 1% user failure rate. This is from clients that receive our specimens, and it's really due to the rigorous QC standards that we've put in place. At the same time, structured metadata really drives reproducibility and supports that cross-study comparability. At BioIVT, we curate clinical data into very highly structured fields and also align that metadata to clinical standards across dozens of different parameters. You can see some examples here. May include demographics, of course, that detailed medical history, treatments, and then all of the specimen verification details as well, including that pathology review. So having deeply annotated clinical and specimen data reduces your sampling bias. It improves that reproducibility, and like I mentioned, it enables cross-study comparisons. So it's important that when you're looking at QC, it's not just a pass-fail. It's really about ensuring the data is biologically meaningful for the question that you're asking. We'll come back to that in some later slides. Really, it's all about pairing the information you need with the question that you have. So as NAMs move closer to regulatory decision making, documentation becomes just as important as biology. And this includes ethical sourcing and informed consent, of course, full chain of custody, and then that compliance with global standards. And so all of these elements together ensure that your data is not just scientifically valid, but also as you move forward in your submission process, it can be defensible and acceptable in regulatory settings. So now that we've talked about quality and variability, the next critical step is making sure that we're actually using the right model for the biological question. And this is what determines whether your NAMS is actually predictive. So I'm running through a few examples here. If your question is perhaps around target expression, you may need systems like matched tumor and normal tissue or primary cells so you can confirm that the target biology, what it looks like in the right context. Or perhaps you're looking at immunogenicity. And then PBMCs and those defined immune cell subsets could become critical for your question. Perhaps you want to look at both healthy and patient cohorts to, again, understand what those real world immune responses look like. For efficacy, certainly modeling variability and again, drawing on that diverse, well-characterized donor cohorts rather than just looking at a single generalized system can become critically important to really understand how different patients respond. And then finally, in a gene delivery scenario, whether it's LNP or viral systems, you may need the right cellular context. So, are you looking at stem cells or hepatic models or specialized primary cells? All of those tied to the organ system or organ target to evaluate that true uptake and function. So the takeaway is each question really demands a specific human model. And when you align those all correctly, you don't just generate data, you're generating data that's relevant, reproducible, and yes, translatable. So we'll close with three simple takeaways. The first being quality at the source drives those confident decisions. We saw that with the variability data, where we saw differences between donors 2 to 4-fold higher, we did not observe that within the same donor. And similarly, in tissue, when selecting the wrong block without the pathology review, can really change the biology that you're looking at or investigating. So if the input isn't right, that translates into lots of problems downstream and incorporates a lot more risk. Second, making sure you have the right partner that's delivering that scientific and regulatory competence. And that means the ethical sourcing, the deep clinical annotation, and of course, standardized workflows that all come together to reduce variability and support reproducibility. Because as these data sets move closer to your decision making and regulatory use, that's where the traceability and consistency becomes really almost just as important as the biology itself. And third, fit for purpose models have to match that biological question, whether it's selecting the right immune donor for an ADCC assay or using matched tumor and normal tissue to understand target expression. The model has to reflect the mechanism you're trying to interrogate. And that's ultimately what is enabling NAMs to be predictive and not just alternative. So if you get that input biology right, everything else, your models, your data, your decisions, they fall into place. And that's what we're trying to drive towards. So with that, I'd like to thank everyone for your attention, and I'd be happy to discuss how you're approaching these challenges in your work, your NAMS workflows during the Q&A portion.